Hello. Hi, everyone. I guess we're just a minute early, so I'm gonna just wait for a second. I think in the short term, for the folks here who are just coming back from your breaks, if you have any questions, please post them in the Ask a Question box, and then Keon and we can take a minute or two to answer those questions from anything you saw today or saw previously in the past couple days. We can try to answer them before doing the next tutorial.
Mm-hmm. Sounds like a plan. It's great to see 170 people joining in.
There's
more
40,
than last year?
There's 40 people live, and I think last year we had a max of 100, around 100 unique people, and I think this year there's 40 people live, which is still great. Yeah. So just for people streaming in, yeah, feel free to ask questions from the previous tutorial or either mine or Dan's lecture components earlier today.
We'll begin the workshop in a couple of minutes, just waiting for people to file in.
Yeah. Maybe while we're waiting, I'll allow Keon to introduce himself in more detail in just a couple of minutes. But I think in the short term, I mentioned previously that Keon was an attendee at last year's, like the inaugural KCNI Summer School, and I'm kind of curious to know, Keon, like how was attending the summer school last summer? How has it like changed the kind of research you did like how did attending the summer school last summer change the research you did over the the past year?
Well, I think like one big thing is you always hear about like the benefits of collaborative and integrative science, like when you attend a seminar or just in conversations with people who do science. But I didn't feel like I had like witnessed that until like, maybe my first conference, but also when I attended the summer school, despite it being virtual. I saw like the string of like science that connected all the independent groups that were working at the KCNI, and I thought that was special. So, on one hand, I felt like, oh man, like the experience I've had up to this point has been pretty siloed relative to like what's going on here. But I mean, people were looking for extra hands, and so I'm like, hey, why not be a part of this really brilliant and collaborative team? So yeah, I guess like that's not much to do with the science itself, but the process of science that really drew me in. Yeah. I also, I like the idea of models, right? I think they can be very helpful at several levels. Like, you could have like what models, which are descriptive, ways to like bring down the dimensionality, the incredibly heterogeneous and messy data that like, exists in the ether. And then, use that to build like how models. So start asking questions about how things are related. And then, I think the ultimate crux would be to develop like why models, like, why is the how model behaving the way that it does? Why does the brain behave the way that it does? And of course, I love the clinical backdrop of all the work that goes on here. I think these are really important questions, and someone needs to use these modern techniques to address them. Yeah, there's so many strengths here.
I think that's a great summary, Keon. Like, I think, certainly one of the defining principles of the Krembil Center for Neuroinformatics is to do science in a less siloed way. And I'm glad that, like, that sort of got through and it inspired you so much that you wanted to do a PhD at the Krembil Center for Neuroinformatics. So that's absolutely awesome. It's a great introduction. So maybe why don't we first, so I'll ask Keon to introduce himself briefly before he jumps into his tutorial, but maybe before that, we can just thank Dan and Sunny for their presentations earlier today.
Yeah, that was like an excellent tutorial, Sunny.
And Keon, do you want to introduce yourself?
Sure. My name is Keon. I'm a recent, well, I'm not even a PhD student yet. I'll be starting in the autumn, just a summer trainee at the moment. But yeah, a little bit about my background. So I went to McMaster, as Srijoy mentioned, and I did a double major in psychology and biology. So I guess like when you bring those two together, you do get neuroscience. So that was the field of my master's, which I also did at McMaster. And the projects that I did there, so like I had a couple projects in my undergraduate where I was really interested in behavioral psychology, just because I liked experimental design and things like that. But then, I kind of wanted to ask more empirical questions, or like have empirical data to work with. So, I was interested in neuroscience, from an earlier age, but, I thought it would be a good, good topic, broad area to do my master's in. And my research was in cats. So for those of you that don't know, the cat visual system is like a classical, very old model that was used to ask all these questions about the organization of the brain, and particularly questions about neuroplasticity in response to experience. So, we had some cats that underwent like some visual manipulations, such as, the occlusion of vision in one eye and seeing how that affects the structure of the visual cortex, but also the expression of key proteins involved in synaptic plasticity. So that's kind of what, that was kind of my foothold in bioinformatics, is because I had some gene expression data that I wanted to analyze. And I didn't want to just do like a couple bar plots with like a t statistic. I came into contact with some like high dimensional analyses, and that stuff was really interesting. I at first I didn't want to use R. I was kind of angry at my supervisor back then for saying, "Oh, you can't use Excel anymore. You need to do R." But it's really given me the freedom to like develop a useful skill. And now I'm like excited every time I get my hands on a new data set and think of the ways that I can break it down and look at it. Yeah, that's kind of a short background on myself. Hope I didn't bore everyone.
Keon, that was a great background. Thanks everyone. And maybe at the end of the session, if people have more questions about Keon's background or Keon's experiences as a student of the summer school from last year, people feel free to stick around or ask them in the ask a question box, and Keon will be happy to answer them. So I'm gonna turn my camera and mute myself. Keon, you can share your screen.
Cool. Yeah, let's dive in. Yeah. So hello everyone. Welcome to the second tutorial. Man, I kind of don't like how I can't see myself, or anyway, as Sunny says, it's kind of like speaking to the void. But yeah, so welcome to the second tutorial. It's a direct continuation of the first tutorial and like a practical application of the data that it produced. And it also ties in some of the clinical interests that reflect the goals of the KCNI as a whole. Yeah, so I'm actually going to touch on a few topics that Srijoy introduced during his lecture, at the benefit of driving home the message and at the risk of being annoyingly repetitive. But yeah, so like we're going to do some analyses in what are called bulk tissue data sets. But what do I really mean when I say bulk? Well, I'm referring to currently the most common form of tissue that's used in transcriptomic studies of the human brain. And well, that's usually an intact frozen chunk of postmortem brain tissue. In order to extract, yeah.
Can I just interrupt you? There's a comment to make the web page just a bit bigger. So could you make your markdown or your HTML?
Oh, yeah, of course. Okay, yeah, please shout out any comments because I'm not able to view.
Yeah, I'll yeah. Okay, we'll do.
Yeah.
One more big, please. Okay, perfect. Okay. Thanks.
All right. Thanks. Yeah. So bulk tissue is just like the whole piece of tissue, usually frozen, almost always frozen, because as Srijoy mentioned, there's some the instances where you're collecting fresh human brain tissue are quite rare. And aside for single nucleus RNA-seq, in most cases, when you want to extract the RNA, you need to homogenize these samples. So in my previous lab, the method that we used were these patented small test tubes that contain ceramic beads. And you put the chunk of tissue in the test tube with some buffer, and you put them in a machine that would vibrate like hell. And then you take them out and you'd have a brain smoothie. So that's what you would use to do your RNA-seq analysis. And because the cells are being destroyed in this process, one might assume that bulk data sets contain no information about the cellular origins of gene expression. But let's consider a simple thought experiment. So here we have like a petri dish, which I'm using to represent a bulk tissue sample. And we can see it's composed of different cell types, which are the different colors here. Just visually, when we look at this, we can see that one major source of heterogeneity on this dish are the proportions of each of the cell types. So in the last tutorial, we used curated single-cell data sets from the Allen Brain Institute to identify genes that are differentially and specifically expressed in different cell types. So logically, marker genes for the same cell type should appear highly correlated, and their expression levels should depend primarily on the proportion of that cell type in the bulk sample. So if, moving over here, if the proportions were to change, one would expect to see that reflected in the expression of the marker genes. And conversely, we can then use the expression levels of markers to estimate the proportions of different cell types in the bulk samples. And this process is what we call cell type deconvolution. And there are many different algorithms for this, but we're going to use one called marker gene profiles. And before we begin, we want to typically have 3 components prepared. We want to have a set of marker genes that we want to have a set of marker genes per cell type that we want to estimate. And we're going to be using the ones that you generated in tutorial 1 from the human single-cell RNA-seq data. Second, you want gene expression that's measured in bulk tissue samples. So this is what you're going to be using to estimate the proportions of cell types, the main data you'll be focusing on. And we'll be using this paper here, which has both control individuals and individuals who were diagnosed with major depressive disorder. And then, most importantly, I'd say you'd want to have a scientific question that you're interested in. And having a question before you start analyzing data is crucial because it'll guide your line of questioning, and it'll also help you to interpret your results and make decisions for where to go next. So in particular, we're going to ask questions about what cell types are changing in response to aging and, again, major depressive disorder. So now I'm going to flip over to, I'm just going to mention a couple of things about where this code lies. So if you want to, here in day 3, I'll just mention this note left by Aaron, who graciously made this whole environment. If you're going to run the code in your Docker, I'd recommend that you update the Git repository using these 2 lines in your terminal. If you're not sure where that is, next to your console, you'll have a little terminal here. You can enter those lines individually and run them to update the GitHub. And then just going there. If you're having trouble with Docker, and I certainly did last year, an option we have for you is to use this link to navigate to this RPub document here, which is a lot cleaner to look at, and probably what I'd recommend, because the concepts are far more important than running the lines of code here. You can always go back and get this working to satisfy your own curiosity. So I'm going to go over to the actual RStudio that's on my local computer, and we're going to first, as usual, load the libraries containing the functions we want to use. This is the package that contains the marker gene profiles functions that we want to use. As Sunny reviewed, Tidyverse is like a collection of really useful packages that share the same syntax. And by syntax, I mean kind of the same language for laying out our code. And I've kind of made an effort to use the language in Tidyverse for this tutorial. And feel free to ask me any questions regarding just like manipulating data using dplyr or other related packages. I'm going to move forward now to loading the data. We're going to first import our metadata. We're going to grab only the metadata that we're interested in. So we have age, we have gender, we have postmortem interval, which is a measure of the quality of the tissue, because the longer this is, the more likely it is to be degraded. We have the integrity of the RNA, pH, and phenotype, which codes the control or MDD individuals. So let's just run this chunk here.
Keon, while that's running, could you just take a moment and say where the data came from?
Yeah. So I'll pull up the paper again. Admittedly, I skimmed over it, but there's a lot of dimensions to this data. So it's a bulk RNA-seq data set. They have both controls and major depressive individuals, and they've measured it. They've measured tissue samples from 6 different brain regions. We've subsampled only those from the prefrontal cortex, so the data is a little smaller and more portable. But there's also a range of ages in the data, so we can look at age effects. And one of the main findings of this paper were there are some sex-specific transcriptional differences within and between individuals with major depressive disorder and controls. So that's one thing to keep in mind, and something I'll mention a bit later once we get there. Yeah, so this is very similar to, first I'll bring up the metadata objects. So yeah, we have the unique geo-accession number here. These are just the identifiers used within the project itself. We have age, gender, and all the other variables that I mentioned. Taking a quick look at the counts matrix. Sunny very articulately described what a count is. So yeah, we're working with counts here as well. The major difference being that each column here now represents an individual as opposed to a single cell sample. Right. I could mention a few things that happen here. We do have to do some cleaning with the gene symbols. So they have some symbols that we don't want in there, or automatically adds periods when they're duplicates of genes. So we're just going to filter those out. And this is a handy line as well. This is when you want to convert a column to your row names, as it would suggest, and just pass the name that you want to have for that. So this was once a column, now it's the row names. And I'll explain a little later why you'd want to have it organized this way in the next chunk of code. You'll also notice that cable will cause some glitches when you have it open. So I'd recommend closing that if you want to scroll down. And as a standard, we want to normalize these counts. Again, because we don't want just broad differences in gene expression that are likely arising from technical variation to be interpreted as biological variation. So we need to do something about that. So we're going to get the relative counts per million. And it's a simple formula like this, where we take the number of reads that are mapped to a particular gene, we multiply it by 1,000,000, and then we divide it by the total number of map reads overall for the chip. Correct me if I'm wrong. Right, back to R. Sorry about that. Let's just run that. And now we can see, well, if you compare it to what was up here, I'm going to preview this again. Things are now relative counts per million as opposed to, reads, counts per million as opposed to the raw counts. Simple enough. We're also going to calculate the row-wise standard deviation. And recall here that the rows are the genes. So if we're getting if we're getting genes with really low standard deviations, like we can kind of estimate that that is arising from some problem with the technology platform, and likely not some biological signal that we want to continue with our data analysis. So we're just going to filter that here. And this also will be important for getting some cleaner PCA results down the line. Yeah, so this function just calculates the row-wise standard deviation. And then we're going to use these brackets here to just subset all the rows for which, this here, just run it. This here is where the standard deviations are stored. And we want to filter rows where the standard deviation is greater than 0.1. We're just gonna, so what this is here is it's a pipe. So what that's doing is it's taking what's created here and passing it along to the next function. So I'm just gonna demonstrate that if you run this just by itself, this row by itself, we'll end up returning a matrix object. But since we want to have row names and such there, we're going to convert it into a data frame. And that's what the second line does, which is then passed to the row name to column function. Rerun that. All right, we're already at the point where we want to import the list of marker genes that we made in tutorial 1. That's here in our data folder right there. So either you generated that while running tutorial 1 by yourself, or I stored it here for you just in case you're using the binder and ran into some issues. But we'll use a line here, read CSV, to read that file in. We're gonna remove the first column because it's just the row numbers, which we don't want. And then mutate is a function which creates a new column. And the first parameter is going to be the name, which we want to assign to that column. And then we use a function called case_when, which is basically a left-right function, where every time this is true, we're going to have what's on the right-hand side of the tilde be what's saved in that new column. So whenever the class label, so let's read in the marker data first. All right, so whenever this subclass label, whenever the class label is equivalent to glutamatergic, we just want to add an abbreviation and make it excitatory. And GABAergic, we want to have a little suffix here for inhibitory. I'm realizing now that when you use dplyr, it's harder to then go back and demonstrate each step because it runs all of it at once. But bear with me. I'll just try to be as descriptive as possible. So what we're doing here is we're going to unite. We're gonna unite two of the columns into this new name subclass label. These are the two columns which we're uniting, and we're going to separate the contents of those columns by an underscore. And if you're more curious about what the other parameters are, I recommend that you use the help, and it'll just tell you what each parameter is used for. So this is a very nice built-in part of our studio, which is helpful. Right, so I united these, and we'll see that there's Na here because, well, astrocytes are neither excitatory nor inhibitory, so we just have an Na there. And right, so we can see the end result here is this new column where we now have the class labels, which were converted from what used to be glutamatergic here to excitatory. And we just grab that and tack it on to the front of the subclass label. And this is something I just like to do because once one goes and looks at figures, it's nice to know, what the class classification of each of these subclass cell types are. Right, and then we're just going to remove any spaces because that sometimes happens. And then we're going to add a new label for those NAs called non-neuronal. Right, last lines. And that's the final output. Moving forward. Now, I'm just going to go back to Crowdcast and see if there's any questions or ask, if the pace is too slow or too fast. I don't see any questions or comments. Srijoy, how's it going?
Keon, you're doing great. One quick comment from me is that when you're showing when you're showing variables in memory, can you make that screen a little bit bigger?
Right. Let's see if I can.
I don't actually know if you can do that in RStudio, but that might be a small help. But if you can't do it, don't worry about it.
Yeah, I don't think I can without fumbling over myself. But yeah, just bear with the smaller tables. Sorry about that.
No worries. It's great. You're doing great.
Great, thank you. All right, so now I'm just gonna do a little paste line. So I just want to see how many matches there are between our marker names, our marker gene names, and the genes that are in our expression matrix, our bulk expression matrix. And there's a good number of matches, so that's good to see. Right, so now I'm just going to grab a vector of the unique cell types we're working with, put them out here. Nice, we have some inhibitory subclasses, some non-neuronal cells, some inhibitory ones. And now we're gonna take this marker data object and we're gonna make a new one, a new object in a list form. So what lapply is basically a special type of apply function, which will iterate across the so cell types here is a character vector of length 19. So it'll sequence along astrocytes, and then it'll endothelial cells, and then excitatory IT, and it will apply a function with each step that it moves forward. So this could be X or anything else, but this is basically taking what's here in the first parameter and passing it to the function, right? So a return line is just basically returning the results of the function. So we're for each iteration of this lapply statement, we're going to take the marker data and we're going to filter it. Sorry for switching back and forth so often, but we're going to filter it by subclass label, and we want to have the subclass label equal the cell type that we're currently on through this iteration. So it'll begin with astrocytes again and then iterate through. And then we're going to pull the gene name, and then we're going to unlist it, and then it's going to be stored as one of the arrays in the list. So let me just run this and show you what I'm talking about. We're just going to add the names too. So actually, before I do that, I'll show you what it's changing. All right, so we now have 19 slots here in the list, and each contains the markers that we generated in the first tutorial. So we can always use something like this marker list, double square bracket at 1, and that will give us the contents there. Now we just want to take these numbers and make them more readable. So names in the marker list are going to be the cell types. There we go. So we went to the trouble of making this because it is the preferred data object, which we're going to pass to the main function, the star of this tutorial, MGP estimate. I'm going to pause for a moment and think about what I want to say. You know what? I'm going to run the code here, and once the outputs are created, I'm going to come back and explain what exactly is going on. So you just tell it what the expression data is, which is our gene matrix. We're going to say the genes are the marker list we just made. And we're not removing the outliers because we had that filtering step prior to this line. And I can answer questions about the other parameters if you're curious. But again, you can use the help function as they're all defined there. Right. So now we have a list, and we're interested in the estimates here. right? So this is not a useful object, so we're gonna transform it from a list into a data frame. That's what this line does. You might have trouble viewing this, unfortunately, but what we have here on the first column is each sample identifier, and then each column is one of our 19 cell types. So we're getting a number here, which represents the relative proportion of that cell type for that sample or individual. I'll mention that these numbers are unitless. The reason for that is that they are a product of a PCA step, and PCA is all unitless. And some of them are negative again because of PCA. But we're gonna make that a bit more tidy by scaling all those values between 0 and 1. So I quickly wrote like a little scale function here. So anything that's passed to scale 0 is going to be this little formula here, which does exactly that, scales it between 0 and 1. And that's for visualization purposes. And add it to our functions in our environment. Now we're going to apply it to our estimations once we remove the sample IDs. And then everything here is just like adding those IDs back. And now we have our estimations in kind of a cleaner format with no negative numbers, a little easier to interpret. Right, so let's start transforming this data frame into another type of data frame, which is best to use when we're plotting, specifically with ggplot. So before doing that, we want to join the metadata for each of these subjects by matching the sample ID to what we have in the metadata. So each row is going to be matched by its ID to make sure the rows are in the correct order. Man, I have a lot of these open, so let me just do some dusting. So first we're going to merge them. And the next step is a pivot longer step. So first I'm going to show you that the merge was successful. All right, we now have all the columns from our metadata, and we also have all our cell type proportion estimates. And what we want to do is we want to have an individual row which describes a particular cell type proportion per cell type. And I'll show you what I mean. We're going to use something called pivot longer. I'm just saying here in the first parameter that I don't want to include the metadata when I pivot the data frame. And I want to take all the names, so all the cell type columns here, all the column names, and put it into a single column called cell types. And I want to take the values that are in underneath them and put it into a new column called cell type proportions. And what this effectively does is it lengthens, it makes the data frame longer, right? So everything's organized like this now. Perfect. So this is generally the type of you usually want like a long data frame when you're passing information to ggplot, because you can quickly filter the data according to what's in each column. So let's say I just want the cell type proportions for control individuals or MDD individuals. I can quickly just filter it by this column or by this cell type, which I wouldn't have done so cleanly previously when those were just column names. So that's the purpose of this. Right, and I'm just going to run a quick line to fix the labels. Yeah, perfect. Okay, so for this section, we don't want to be lazy. We want to make sure that these marker gene profile calculations that we made make sense or look good. So we're going to do some quality control. And the code here is long and detailed. So what I'm going to do is I'm going to run it first, and I'm going to loop back if there's additional time to explain what's going on. But it's much more powerful to see what the output plots are first. So that one and this one. Okay. So there's some things to be said about this. So what we have here are the number of gene markers per cell type. And you'll notice there's quite a bit of variation here. And there's two main reasons for this. So one reason is that in the Seurat tutorial, that cell type was more rare. And just by how the statistics work, it's less likely that you're going to detect differentially expressed genes for that cell type relative to all the other cell types combined. So that's one reason why some cell types might have fewer markers as an output. The second reason is the number of markers that are displayed here are actually different. They're actually different from the number of markers that are here in this marker list. And the reason for that is not all of these markers are retained by the MGP estimate function. And the explanation for this is we want to use an upregulation of cell type specific genes to deconvolute the proportion of cell types in a bulk sample. But we only want to retain those genes which are being that are highly correlated with the cell type proportion and that are not being regulated by something else. So one way that this happens is that, let's see, what's the best way to explain this? Okay, so Sunny gave you an intro to what PCA is. So PCA is a method by which you create a series of orthogonal vectors that iteratively capture the greatest variance in your data set, right? So the first vector will capture the greatest amount of variance, the second one, the second greatest amount, and so forth. And they're all perpendicular to one another. So the statement that I made when I began was that one of the main sources of heterogeneity within a bulk tissue sample is the proportion of different cell types that are there. So we would expect that the first principal component, which describes the greatest amount of variation in that bulk sample, is representing the variation from changes in cell type proportions. So effectively, what this deconvolutional algorithm is doing is it's seeing how many of the genes are being aligned with that first principal component, and then it's using those principal components as a measure of relative cell type proportions. I can circle back, and with the help of very talented TAs and Srijoy, who know a lot more about this algorithm, because Srijoy was actually part of that paper which published this algorithm. And they can help answer some questions there. But what we want to do is we don't want to keep markers that are being regulated, for example, by major depression. We just want those that are telling us information about the cell type proportions. So any of these genes, any of these marker genes, which are oppositely correlated with the majority of all the other markers, are dropped. And this is because they're likely being regulated by something that is not a cell type proportion. Okay, there's a lot there, and I can always circle back and explain more. But yeah, so that's why some of these cell type numbers are different than the number of the number of marker genes that initially went in. Some of them are dropped because they're non-correlated with the other markers, and this is a quality control step. We also have a plot here. Right, yeah, it's telling us how much of the variance explained in PC1 is attributable to these cell type proportions. And the line here is saying, you know, that we want more than 35% of the variance in the first principal component to be aligned with what these markers for each cell type are explaining. So some fall below that threshold. which might mean that they don't have an good markers. Or there could be a couple of things. The genes which represent those markers could not be well represented in the bulk data, or the marker list might not be so good because they're rare cell types. But yeah, we're not gonna drop any of these because of this quality control metric here. We're gonna keep all of them, and we're gonna move forward with our analysis. Right. So now let's start integrating some of like the questions we asked at the beginning. Like we want to see the relationships between cell type proportions and disease status or age. So what I'm doing here is I'm creating just like a vector of cell types we might be interested in. Somatostatin cells are definitely have been associated with major psychiatric disorders, and we'll hear about that on some later days. So that's one that we're interested in. We're also interested in oligodendrocytes because they're often lost in psychiatric disorders as they're sensitive to metabolic deficiencies or stress, as well as oxidative stress. With a caveat I'll mention in a moment. Right, so vector there. And now I'm just initializing a list where I want to store the plots I'm going to make in this for loop. Here's the variable that is going to store which iteration we're on, typically called I. And I is going to sequence between 1 and the length of the genes that we have that we're interested in. So 3, between 1 and 3. And then in the first slot of this list, it's going to store the output of our ggplot, and we're gonna take our data set, our long data frame that we made earlier, and we're going to filter by first excitatory IT cells. We're going to define the aesthetics that we're interested in plotting. So we're going to have age on the x-axis and cell type proportions on the y-axis. And then we're also going to separate, we're also going to color our dots, which represent each individual by their disease status. Let me just run this whole chunk. Okay. So this might be hard to see. So what I'm going to do is go here. So again, we have excitatory IT marker gene profiles or cell type proportion estimates. There's scores listed here on the y-axis, and then the age of the samples on the x-axis. And the blue dots here are MDD individuals, and the red ones are controls. We can kind of see that they're diverging, where for some reason the excitatory IT cells seem to be increasing with age in the control individuals and decreasing in the MDD individuals. Pretty similar trend here for inhibitory SST. This is something that may be unexpected, and I can give a couple reasons why. So we'll note that like there's a decent amount of spread for these points here. And one is because we're using only, I believe, 45 individuals. Again, that was for the portability of this tutorial. There are a lot more in that data set, and we probably get some tighter associations with a higher N. But there's also a significant amount of subject heterogeneity here. So we also, have a wide range of ages between 20 and 80. Yeah, that's also contributing to how variable the individuals are. So yeah, the data is noisy, and it might be hard to parse some of these signals. So one option we have here is to use linear models. We actually use a linear model in making this ggplot. So if you notice here on this line, geom smooth is to plot this line tracking each of the conditions. And the method we're using is LM for linear model. So the way that the algorithm is plotting this line is that it's taking the distance from each point to the line, which is called a residual, and optimizing that. So minimizing it as much as possible to get the best fit. All right, which is in essence what a linear model is. And then we basically have a slope and y-intercept. So y equals mx plus b, as everyone knows. So what we're trying to predict here is the cell type proportion given sample age, and we can describe that as an equation where the slope of this line is the beta, and then we have a number here for the y-intercept. So what multiple regression is this very principle. But if you want to imagine it, all we're doing is we're adding another dimension of data, right, to account for that when we're optimizing our linear model. So it's akin to adding another axis here, another perpendicular axis, which would, for example, be the values for PMI. So now each point is being plotted in a three-dimensional space according to three variables, and the steps for minimizing the residuals is basically the same. It's just happening in 3 dimensions. And it gets a bit more complex to think about when you add several more parameters to your or predictors to your linear model, but the principles remain the same. So what we want to do here is we want to include all of these different subject variables, such as the disease status, the gender, the pH of the brain sample, the RNA integrity, the PMI, and the age of the individuals, to kind of hold them fixed so we can look at individually the effects, the contribution of each term to the changes we see in the data. So there's basically, it's going to calculate a coefficient or a beta for each of these terms, and then we're going to plot the beta to see what the contribution of each term is. I'm going to zoom up just a little bit so I can see myself. There we go. Right, so we're taking the same long data frame from before. We're going to group them by cell type. So we're going to perform this analysis within a cell type. So when you use group by, every line thereafter is happening within a certain group. All right, and then we're going to use the LM function. And we're going to try and predict the scaled cell type proportions as explained by phenotype, gender, PMI, pH, RIN, and age. These functions here are just to clean up the outputs of the linear models so that we can extract them a bit more easily. We're going to ungroup the data, so now we're going to apply everything to all of the rows instead of within certain cell types. And we're going to apply an adjustment to the p-value for multiple comparisons using the Benjamin-Hochberg method. Right, and we're just going to recode some of the variables into more interpretable names. And let's go back and run this so it's a bit more clear what's happening. Great. All right, so we get an output that looks like this. So for each cell type, we have a term. So we have the intercept, of course, and a beta coefficient estimating the magnitude and the direction of the term here for MDD. So all the changes here are relative to control. So this is the effect of having MDD, and that's the estimated beta. There's an error associated with it, p-value and adjusted p-value, and so forth. I also added the class information so we can clean up our ggplot to be within each class. And this is what I mean. All right, and I'll switch to this, which is more viewable. Right. So here we're plotting the betas. The beta effect for an MDD designation for each of the excitatory cell types, inhibitory and non-neuronal cell types. And what we can see from this is the error bars for the standard error are huge. This is not surprising to me, given how small the data is. Ideally, again, we would include more samples. But it's also a noisy data set. So there's a lot of variability that's being driven by age. And actually, as we scroll down, we see age has kind of a more pronounced effect, or at least a more, I guess, less more certain effect. I don't want to use any language that's statistically incorrect, but let's say that the age seems to be driving this a bit more than the disease status, which makes sense. The brain goes under some pretty big changes between these years. It's just some things that we can see is that with age, we see an increase in oligodendrocytes, but it's important to look at this data in a holistic way, because if we're seeing a relative decrease in neuronal types pretty much across the board, then it's not surprising to see that non-neuronal types are increasing as a result. So it's a two-way street. This could be a loss of inhibitory and excitatory neuronal types, or a relative increase in neuronal types. So there's just some caution when interpreting these results. Right, I'm going to check for questions because we're at the end of the tutorial now. I don't see any questions in the chat.
Keon, you're doing great. Can you go back to the bar graphs and just maybe just spend a couple moments sort of explaining like what do the directions mean, especially maybe starting with again the age graphs and then going to the MDD versus controls. Like what is, especially for the MDD versus controls, like what is reference? Like what does a negative beta coefficient mean?
Yeah. So again, like beta is basically like the slope, right? So if we're going to conceptualize these as like plots similar to this again, we would expect a negative relationship between the two variables when we see a negative beta. So the way to put that into words is with an increase in age, we'll see a decrease in excitatory layer 5-6 IT CAR3 neurons. Conversely, as age increases, so does the relative proportion of oligodendrocytes. Carrying that over to disease status, we see that since it's a binary difference, if you have MDD, you're more likely to have a depletion of excitatory layer 5-6 and P cells relative to controls. Whereas with some variation, some mixed results, you're more likely to have excitatory layer 4 IT cells relative to controls as an individual with an MDD diagnosis. Is that sufficient?
Yeah, that's great. That's great. Yeah, that's exactly what I was looking for. Thank you.
Great. Yeah, that is the end of the tutorial. I hope I demonstrated, despite our results, that there's definitely a lot of utility in identifying marker genes, because it allows, although correlational, these types of connections to very high-level phenotypes such as neurological disorders, neuropsychiatric disorders. And as the amount of data increases, and the sophistication of the algorithms increases. Well, that's not necessarily, the last part is not necessarily something you want. Like PCA actually does an excellent job, and it's not certain that a more sophisticated nonlinear algorithm will capture better differences. But yeah, as a whole, it's really interesting to connect single cell RNA expression to a high level phenotype such as major depression.
Keon,
How do I stop sharing my
it.
screen so I could see?
Okay, so now you're still sharing your screen.
Can I hit close video? I'm worried about that. Okay. Oh, there we go.
Yeah, great. Yeah, that was great. Thanks, Keon. Let's just do a round of applause. Yeah, that was great. There's one question in the chat from Fernanda. Do you want to see if you can answer that?
All right, to read it out, there are no subjects with MDD more than 70 years of age. It impacts the graph, but would that impact your model? I'll be honest and say I don't necessarily know. I mean, like that, things like that is one thing that using like linear, like multivariate linear models and generalized linear models tries to quote unquote control for. But you can never truly quote unquote control for like the range of your data, or how many subjects you have within each group.
Yeah, like basically, like that was an excellent answer, Keon. Like my understanding of it is that exactly with these linear models, we're trying to address the correlations between various variables and sort of explain away variance due to age, and then once you've explained away some of the variance due to age, then you're sort of left with like maybe your condition or your contrast of interest, which would be like MDD versus control. Subjects with MDD versus subjects who are have a control designation, but there's no amount of statistics that can address or help poor experimental design. So, for example, let's imagine the experimenters didn't do this, but let's imagine they did an experiment where everyone who's a control subject was in their early 20s and everyone who's a subject with MDD was in their late 80s. So we call that in bioinformatics, or in statistics, we would call that complete confounding between the cases and the controls. And you wouldn't be able to tell apart what was the difference that was due to the ages between the different subjects versus the difference that was due to, having major depression, being diagnosed with major depression.
Right.
Yeah, so typically when people run these experiments, they try to design them well so that they have an equal, they have a equal variation between control groups and equal variation between the subjects with major depression. But certainly in that study, having subjects with major depression who sort of span the range, the natural range of aging from like, age 20 to 80, that's a huge range, right? Like, it might have been better to try to restrict subjects to a more narrow range, but yeah, who knows? We didn't collect the data, but as bioinformaticians, we often reuse the data of others, and we're just beholden to decisions that were made by others, and we try to analyze what we can. But that's an excellent question, Fernanda. More questions for Keon? And I'm sorry to sort of interrupt with the.
No, that was a great explanation. Yeah. Yeah, they're like not necessarily websites, but like there are papers, right? Largely methods based that like present their deconvolutional algorithm, and then often they have a data set which they demonstrated on, and if they're partial to open science, they're going to publish those markers, and they likely will because like the performance of their algorithm is going to be reflected in the markers. So yeah, there's several papers that that publish their markers. I guess the caveat is like what you have to kind of like dig to see what kind of data it went in, and like convince yourself that like it's going to be relevant to your question. Yeah. There's some papers that even build lists of markers without using single cell data at all. So they use entirely computational methods across like hundreds and thousands of bulk samples, and using the same like axiom that I mentioned earlier, that you would expect natural variation in between bulk samples. And if you can capture that and associate the principal component, which is most correlated with traditional markers of certain cell types, like GFAP for astrocytes, although like that's not so. It turns out it's not that great of a marker, but yeah. So you could just like see like which principal components are most correlated with traditional markers that have like an experimental basis, and then you can just grab all the other genes which are correlated with that vector. So yeah, you don't necessarily even have to start with single cell data, but it's definitely better to do so. I think you're muted, Shuja.
Yeah, thanks, Keon. Sorry, I just linked to a list of mouse-based marker genes that you could use exactly for that purpose. Mm-hmm. If you have any questions sort of navigating what I just sent you, please feel free to ask, and I'm happy to help you sort through it. Yeah, there's number for many cell types, like there's many lists of markers. I think, unfortunately, sort of behind the scenes, we sort of presented this nice set of tutorials to you. Like we spent a lot of time in my lab and in Dan's lab arguing about marker genes and what's a good marker and what's not a good marker. And it certainly affects the outputs of results like the tutorial that Keon just showed you. There's a lot of thought that goes into these, and yeah, a lot of the secret sauce of bioinformatics is in making decisions like that. But anyway, in any case, that was a bit of a digression. The research I just pointed you to contains a list of high-quality mouse microglia genes that you could use for your purpose. More questions? Maybe we could open the floor to just more questions in general, sort of like questions in general and broadly about like, let's say the practice of bioinformatics or the practice of doing bioinformatics in neuroscience, or if you have questions, more questions about, either for myself, Keon, Sunny, and Dan, we can likely invite Dan and Sunny to join us, and you can feel free to ask questions.
Even if you have like a question you're not sure about, just throw it out. That's what I tell myself. Like I always feel like a little inhibited, like with these types of things, because there's a disconnect between the screen. But yeah, I'd be happy to answer any and all questions.
In the meantime, I'm putting together a poll.
Cool.
My poll is, are you more likely to use gene expression data now than you were before? Yeah, we'll just wait a couple minutes.
Trying to think of anything interesting I could share, but
okay. No need to belabor the point. Like, what are the limits of measuring RNA? Why haven't we solved mental illness? Yeah, that's an excellent question, Dan. Keon, do you want to take a crack at that, or do you want me to attempt
Dan,
it?
That's like a real gutting question, though. I'll let you go first.
Yeah, I don't even know where to start, but yeah, clearly, like, I think a lot of mRNA, and I think it's like, I'm not a very religious person, but I think but I think like RNA and like the beauty of being able to measure RNA ubiquitously from many cells and many conditions relatively cheaply today, like I think it's like, kind of one of the gifts from our creator. And so, and again, like that same magic is what enabled these awesome mRNA vaccines. So I think, but that all being said, it's what you're measuring is, because most methods for measuring mRNA are destructive, you're measuring mRNA from a cell or from a tissue at a snapshot in time. So a lot of so for example, for the data set that Keon was talking to us about, that was measured from the post-mortem brain. And so those people are, they passed away, they gave their brains to science, and that was studied. Afterwards. So we can't, unlike other types of data that you'll hear about, in other days of the KCNI summer school, because measuring mRNA is destructive, and because the brain is like, we can't easily biopsy it, ethically anyway, that it's just challenging to use mRNA to sort of answer all the questions that we might want to know. I firmly believe, like, if we were able to biopsy brain tissue as easily as we could in cancer, progress in neuroscience would be a lot further than it is today. But unfortunately, we can't do that for ethical reasons, and that's probably for the best. So
I'd say some like oh, sorry to interrupt you.
No, please go ahead.
I'd say some of my most sobering moments are like when I come across a paper or generate my own figure that shows a correlation between like mRNA expression and like the expression of those same proteins for which they code. And like sometimes you can't get it above like 40 or 50%. So there's a lot of levels of analysis, right, between like the complexity of like a mental health phenotype and the biology that begins with genes and then goes to mRNA and then goes to proteins. But I think like, and again, like there's also the limits of like working with postmortem tissue in most cases, and the limits of like only having a single temporal snapshot of what's going on in a cell. And the assumptions that need to be made to have these analyses work is that the biological processes that are upregulated at that moment in time are reflected accurately in the mRNA, that no like technical and subject variability is like destroying that signal. And then, those changes in expression are carried up through like the biological chain through proteins into like cell function and so forth. And, I think the technology might say this, right? Just to mention like patch-seq again, that last couple slides that Srijoy showed. Once we can like convince ourselves that the associations between mRNA and morphology and electrophysiological properties within a cell and then eventually within a person are real, then we can build better predictive models. I'm rambling. Go ahead, Srijoy.
I have an analogy, and you have, especially when we talk about like more like a theme throughout the summer school is that there's different ways and different levels of investigation of the brain, and the theme for today, or the theme for the early days, is that we're sort of at a more molecular level, and so we have incredible detail with which to study the molecular level, but most of those methods are destructive. So whereas like as you get sort of go higher up the chain, like if you think about brain imaging, it's not destructive, and so but it's lower resolution. So there's a trade-off there. Like you don't have to require the death of a person in order to study the thing that you want to study. So that's good, but then it's lower resolution. So a lot of what we're trying to do at the KCNI is try to integrate or bridge these levels. And as Keon said, so how can we take these mRNA measures that are destructive, but they're incredibly, they're now becoming ubiquitous, assuming that we can get the tissue needed to run them. How do we relate that to more physiological measures of cells and tissues? I just thought of an analogy, and I thought I'd try to pilot it here. So imagine the brain is like a car, and we want to understand how the car works. And these measuring mRNA is like taking the car apart and measuring, like sort of counting up the elements that are present in the car. And sort of through some of the cell type based methods, we can sort of turn those different elements back into, we could say like, oh, well, it appears like the car has, this car has leather seats, and that other car, well, the leather seats are a little bit worn. That's kind of what we're doing. Whereas like some of these more, think about brain imaging or physiological measurements of the brain that are not destructive, it's like watching the car in motion, right? But you're not, it's fundamentally different than taking the car apart and measuring all the pieces one by one. But clearly, like, well, I don't know how much you learn about the function by looking at leather seats, but you could learn something about the engine from more molecular based methods, and that might inform how fast the car can go from, like, say 0 to 60 kilometers per hour, 0 to 60 miles per hour. Yeah. So again, like these, we firmly believe that these are all complementary approaches. I'm clearly biased. I have my favorite methods, but that kind of comes from my effectively my anal retentiveness and my desire to want to understand like the thing at the most granular resolution possible. Sunny asked, I wonder if there was a day during the summer school where we talked about integrative research more explicitly. I bet that's probably on the schedule, but I don't actually know. Maybe we can sort of bring Erin on if she's around, and maybe she could speak to that. So I'm gonna invite Erin on screen so she could sort of ask about, are we good? Yeah. So we're gonna wait a moment for Erin to join. And if Erin can't join, that's fine. If someone has the answer as well, feel free to post it in the chat. Hint, day 8. Oh, okay. So
I think he was asking.
Yeah. Okay, so I guess there is a day, and cool. So we'll talk about it more explicitly. Okay. So yeah, I think that's a good question. Fernanda asked another question: if we increase the number of genes used to discriminate cell types in the first tutorial, will we improve the detection of other cell types that were not detected well here? Yes, absolutely. Absolutely. But there's a trade-off because or in practice, there's a trade-off though. And also the cell types that we're trying to distinguish, like they're not that different from one another. Like well, depending on who you are, like, or I wish I'd mentioned this more during my session, but like the cell types are organized inherently in a hierarchy. And so, and just like a natural taxonomy of species, we have different types of neurons and different subtypes of neurons and different subtypes of neurons. But the differences in terms of gene expression of the different subtypes is not that large, but the differences between, like, say, a neuron and an astrocyte is huge. And so there's many genes that will distinguish your generic neuron from an astrocyte, but there's few genes that will distinguish, say, neuron, PV, parvalbumin neuron subtype 1 versus parvalbumin neuron subtype 2. And that's a challenge, or that's a limitation in using these types of data sets to infer things about cell type proportions. Yeah, that's a great question, Fernanda. Erin just posted a link to the post-workshop survey. Again, just as a plug, it'd be very helpful for all of us if you guys would fill out the post-workshop survey, and then we'll be careful, we'll be mindful to use the feedback for future iterations of these tutorials going forward. Maybe we'll just wait one more moment for more questions. These are really great, by the way, and they're forcing us to think. Thanks, Fernanda. Okay, so why don't we thank Keon and Sunny again? Yeah, that was really great. As an advisor, and I'm just so proud of both of you. And you guys are becoming great instructors, and it's really awesome. So it's great to see, and thanks so much for leading these tutorials, putting together tutorials and leading them. I really, really appreciate it.
My pleasure. It really is. I hope everyone learned something. I learned something in the process of putting this together. So
thanks.
Only gains.
Okay. So why don't, can someone post the link to GatherTown in the chat, and then maybe I will end the session, and then people can gather in GatherTown. And thanks everyone for attending, and thanks for the questions. This was it was nice to take a day away from doing science and teach this stuff to everyone. Okay, thanks.
See you tomorrow.